Okay. Well, thanks for this opportunity to tell you a little bit what we have been doing in the recent months, and in fact 2 or 3 years in total. Although I will like first to introduce a little bit the lab first, particularly the two groups that have collaborated in this work, and the people who've done the job, Galal, Alexis, and David, and also with the help of Marta, have done the experiments in yeast. But I also show you some data that we obtained in mammalian cells, and they were obtained by Eva and also Monica. And Karma and myself were just trying to be positive every time that something went wrong or something had to be modified in order to understand a little better what we wanted to know. I will not say anything about the title because I prefer to go straight to this introduction, but I can just let you know what are our main interests. And in fact, during many, many years, we focused our attention to G1, and we used most of the time, we used the budding yeast because the molecules that control this transition, the G1 to S transition, are pretty much conserved. So yeast is very easy to manipulate, as you know, and experiments can be planned very, very fast. And the main idea that everybody accepts regarding cell cycle entry is that we have a particular level of CDK cycling activity that eventually, while the cell grows in G1, we'll be able to start phosphorylating wave 5, which is the PRB homolog in our cells. And that will lead to transcriptional wave that will include other cyclins that will help the CDK to phosphorylate those factors at a higher level in a positive feedback loop. And in this way, making start or cell cycle entry a switch. But very early, when these cyclins were characterized, we learned that the level of the cyclin, of these cyclins, G1 cyclins, do not change much throughout the cell cycle. So in fact, they're called cyclin because structurally those proteins have all the attributes of other cyclins, and they activate the CDK, of course, but the levels don't change much. So the question remained, and it still remains in a way, that, okay, so what are the mechanisms that using pretty much constant levels of the CDK and the cyclin throughout G1 will help the CDK or allow the CDK cyclin complex the phosphorylation of the factors at the very beginning of a start. And we also, in our work, one of the things that we learned is that the CDK cyclin complex accumulated progressively in the nucleus during G1. And this was because the CDK cyclin complex somehow is retained at the cytoplasm in early G1 cells, and is only released so it can accumulate in the nucleus in late G1, very close to start. And for this release, we also learned that chaperones were playing very important roles. Of course, at the beginning, the first time that we found chaperones in our IPs, we thought, okay, another contaminant. But then we finally decided to give them a chance, and we learned that the role is limiting. And in a way, all of the processes that require chaperones in the cell are competing with this particular complex. So depending on the availability of the chaperones, the complex will tune or will be tuned in order to accumulate in the nucleus or not. Among the connections that we've been able to establish between chaperone availability and physiology, let's say. So we work with connections with cell size. In other words, how would chaperone levels or chaperone available levels, how would they transmit the information to the cell cycle regarding cell size but also growth, and finally stress. And today my talk is going to focus particularly on stress at the very beginning of the cycle, of course, because we know quite a lot about how stressful conditions modulate or regulate the ability of the cell to progress through the cell cycle. But in this talk, I will refer to the mechanisms that act, or that we think act, at the very beginning of the cell cycle. We first started by, and that was many, many years ago, from a screen, or this story started from a screen. We were looking at hyperactive CDK mutants. In yeast, having a hyperactive cyclin or hyperactive CDK usually produces a reduction in cell size. And that's because the cell enters prematurely in a start when the cell has not yet reached the critical size. So what we did was just to mutate CDC28, which is the only CDK in. But in yeast, we placed the cells here on top of the gradient. Then after a while, we just picked up the cells on top of the gradient, the smaller ones. And we just repeated this 3 times to finally sequence the CDC28 alleles that were present in those small cells. And we've obtained, of course, a bunch of mutations, but they could be grouped in 2 different sets. On the one hand, this is the CDK, the 3D scheme of the CDK. And on the one hand, we had some mutations very close to the region of the CDK that's bound by the cyclin. So we were not interested in those ones because most likely those could stabilize the interaction between the CDK and the G1 cyclin, which is particularly unstable, right, in all cells. But instead, we focus on another group of mutations that were clustered right in the other side of the CDK. So any function that we perform by this part of the CDK would be perfectly compatible with cyclin binding. And all the mutations that we found, or most of them, affected positively charged amino acids, suggesting that these small cells were small because they had this hyperactive CDK, because the CDK was not interacting properly with a negative regulator. And this was, in fact, our objective, to identify negative regulators of the CDK. So then the next step was just to characterize the interactome of the wild type and the mutant proteins. And among the candidates, we initiated our work with V7. I will not talk about this because this has to do with the retention mechanism itself. But then Galal, who was the one who did this part of the work, he noticed that there was another candidate that could be interesting. That was a putative RNA binding protein. It had not been characterized yet. But there were some data that would locate this protein in the stress granules. And since we were interested in the coordination between cell cycle entry at the very beginning of cell cycle and the stress, we decided to characterize further this protein. The first thing that we did was just to confirm that it was a negative regulator, so cells that were lacking V8 were smaller, as expected. If we overexpress V8, well, we observed an opposite behavior, so cells would become larger. And it was important for us to know or to tell whether these effects require CLIN3. And as you can see here, if we get rid of CLIN3, of course, if we get rid of the G1 cyclin, of course, for the cells, it takes a lot of growth to be able to trigger a start, and they become very large. But in those cells, if we got rid of V8, we did not obtain a significant reduction in cell size. In other words, whatever V8 was doing in those cells required the presence of CLIN3, at least regarding these simple phenotypes. Then when we compared several protein groups and just asked, okay, do they have anything in common? So we just looked at the proteins that have been characterized as CDC28 interactors, physical interactors, proteins that had the ability to bind RNA, proteins that were upregulated by particular stress, in this case, HA, and proteins that belong to the stress granules. Just one was complied all the properties, and that was V8. So, well, we decided to go ahead because if it had a role coordinating cell cycle and stress, particularly in the stress granules, that could be particularly unique. By the way, perhaps I forgot. Of course, we baptized this protein as V8 as soon as we knew that cells lacking V8 were small. Just following the saga. We means small in the Scottish, but some of you may know that Scotland was the place where many cell cycle division mutants, or cell cycle and division mutants, were obtained in yeast. All right, so let's go for the stress granule. We also know that the stress granules are formed whenever the stress condition is strong enough to affect translation. And the general idea is that mRNAs that are not being translated, but still bound by proteins that naturally would bind these mRNAs, will be able to form multivalent or to create multivalent interactions among them, which would finally lead to a liquid-like structure, which is called the stress granule in this case. There are different types of these RNA or RNP condensates. There are P bodies also, but they will only refer to stress granules. So something that appears and disappears only under stress. Other RNP condensates are formed under normal conditions and they have a specific role. Okay, the first thing that we did was to characterize the interaction between V8 and the CLIN3 mRNA, because if V8 had RNA binding domains, could it be possible that it bound CLIN3? And that was the case. I'm not going into detail into the data because I'd like to spend some more time at the end with the fact that we are able to show that this is a bistable system. And we also learned that V8 interacts with PUB1. PUB1 is, let's say, a central protein in the stress granule formation, and it's a poly-A binding protein. It's a protein that binds, well, almost all RNAs, and as long as they have a poly-A tape. And PUB1 has an important role in the stress granule assembly. We also went ahead and asked about their colocalization, and that's what we found. So the V8 was found pretty much in the same granules where we could detect the CLIN3 mRNA. And this colocalization required the presence of V8 and also a little bit with V8, but especially V8, which is the one that we were interested in this work. We also then asked whether V8 would recruit the CDK here, because of course V8 had been identified as an interactor of the CDK that depended on that positive patch on the C lobe of the CDK. And yes, this is what we found. We found that the CDC28 was heavily recruited to S granules under stress conditions, not the whole protein. So we figured out from the images that perhaps 50%, around 50% of the protein accumulates in the stress granules under stress. And again, this recruitment depended on V8. So in cells that were lacking V8, we did not see that much localization of CDC28 in granules, now using PUB1 as reporter. Then we finally, we wanted to know, okay, what for, right? What would the CDK be recruited here? Particularly if it's only a fraction of the CDK, because if the whole CDK or most of the CDK were recruited, you could say that these could be used by the cell to prevent its activity or to inhibit its activity, so preventing cell cycle entry and progression. But we just found that 50% of the CDK, perhaps some especially sensitive steps of the cell cycle to CDK concentration might be affected, but well, it was not reasonable. So then we asked the opposite question. Okay, could it be that the CDK regulates somehow stress granule dynamics? Well, stress granules are formed very fast in G cells, so it's difficult to tell if something may help a process that is already very, very fast. However, the dissolution, stress granule dissolution, when you get rid of the stress, when the cell is placed under normal conditions, back to normal conditions, that's a slower process. And this is when you can ask and tell whether something is affecting as stress granule dynamics or not. And this is the experiment. So here we have the dissolution. This is time zero is the time that we place cells back to normal conditions. And so we start with maximum stress granule formation, and this slowly disappear. And after a couple of hours, you see almost no stress granules in the yeast population. However, if we knock down CD20 activity using this thermosensitive allele, then we saw a nice decrease in the stress granule dissolution rate. Overexpressing CLIN3, well, we observed a slight increase in this dissolution rate, again confirming that perhaps, yeah, CDC28 had something to do with the stress granule dissolution. Then we also use another approach where we, instead of affecting the CDK, we were able to get rid of all G1 cyclins. And without G1 cyclins, the cell rests in G1 with no CDK activity. So it's another way to downregulate CDK activity, but now not affecting the CDK itself, but just the partners. And again, we found pretty much the same result. So if CDK was off because there were no cyclins around, then dissolution was strongly delayed compared to the conditions where we had the CDK on. Yeah. So somehow the CDK, directly or indirectly, was inhibiting stress granule stability, or was fighting against the stability of stress granules once formed. All right. Then we also took a look at mammalian cells, as I said, and yeah. And then we just pinpointed equivalent actors. Caplin-1, we found that Caplin-1 is able to bind the cyclin D mRNA. Tia-1 is the homolog already known, or for PAB1, so there's no problem here. And we also found that the Caplin-1 and CDK4 interact with each other. So the question is, okay, now what was the physiological role of such a mechanism where we have this recruitment of both the mRNA of the cyclin, so the cyclin cannot be translated, and the CDK. And the first thing that, well, occurred to us was that, okay, during the cell cycle, CDK activity changes a lot. So in G1, the CDK activity is pretty low, and it increases to very high levels in S phase and then later on in mitosis. So could it be that this would be a mechanism to modulate the stress granule formation depending on the cell cycle phase? So then we compare, in both cases, we compare yeast cells and U2OS cells just by looking at G1 cells or cells that had entered the cell cycle. And in both cases, we saw that in G1 cells, the stress granule dissolution was, again, decreased. Right? So, well, perhaps if a cell is in G1, having or staying under the effects of a stress is not as bad as if the cell were right in the middle of DNA replication of mitosis when the stress conditions came in. So that would make sense. But we also wonder whether, I mean, the dual play of the dual role of the stress granules in inhibiting translation of the cyclin, on the other hand, and having the CDK playing a negative role in the stress granule formation, we wonder whether this double negative feedback could give stress granules the property of being a bistable system. So I will not go enter into the math, of course not, but we use a very simple scheme. This is just pretty much the same as we saw in the last slide, but just using some formal terms that we can follow and define in a mathematical way. And this is the result of just plotting stress granule formation against the stress using these equations. And we have this S-shaped curve. Just a word about S-shaped curves. I mean, you will recall for sure when we learn, for instance, parabolic functions in high school, that, well, they will give you two different solutions, right? And in those problems where you would try to wonder or to know the age of someone, having as data different sorts of pieces of information, some of them raised to the square, and then you would end up with two solutions. Okay, many times one positive and the other negative, and you would ask the instructor, okay, what do we do now? And the instructor would say, well, choose the one that makes more sense. I mean, don't worry. I mean, never mind, right? Well, in here, although we have an S-shaped curve, all possible solutions have an important meaning in the physiology, in the functioning of the system. In other words, if you have the system off and you start increasing the stress, if you follow this curve, that means that you can increase the stress quite a lot, and yet the stress granules will not be formed. However, once you go over a critical value, then the system will quickly jump and produce lots of stress granules. However, if you start from the other side, so if the cell is already under a stress condition, that means, well, that is with lots of stress granules, and then you get rid of stress slowly, then even if you reach the same stress level as was used before to produce stress granules, in this situation, the stress granules will still be in the cell until another critical value, which lower compared to the first one, is reached. So that's what helps the system to have a memory or to behave differently depending on the previous state. That's called hysteresis. Yeah? And of course, if we had no inhibition at all, we would have this sort of Michaelian kinetics, just producing lots of stress granules just with a little stress. Okay, so the usefulness of these bistable systems is quite obvious. I mean, they help the cell to prevent the stress granules under conditions that do not merit all that effort, okay? Particularly when the stress, so when the stress is not that high. And then if the stress has already been stressed, so it will not revert easily. So the stress will need to decrease very much. So the cell takes the chance to say, okay, the stress is now low enough to revert the whole thing. How do you do these experiments? Well, this is just the results of the model. Sorry, am I running short of time or?
No, you're good, but it would be nice to have some time for questions after. So maybe
okay,
five more minutes.
No, in 2 minutes. Yes. Thanks.
Okay, perfect.
Yes. Well, this is the output of the model. And what you do in your experiments in real life in order to know whether a system behaves in a bistable way or not is to execute or to perform the experiment in the forward mode that we call in the reverse. Let me go ahead with experiments. This is the experimental data. So in the forward mode, we just start with cells that are not stressed, and we just apply different levels of stress. And then we just follow the number of stress granules in cells. Now, for the reverse experiment, we do the opposite. So we just start with cells that have been stressed to maximum levels, and we start decreasing the level of the stressor, in this case, sodium iodide. And we also record the number of granules. And as you can see is here in this kind of dual experiments where you can tell that a system is behaving in a bistable manner or not. Okay? All right, so let me just move on. We repeated the same experiment, but now with this strain that we can fool around with the levels of cyclin, so we can have the CDK either on and off. And well, the experiments were similar to what the model was proposing if we decrease the ability of the CDK to work. So this result means that even though we have no CDK at all, the model still would say that, yeah, there's something. So that means that there should be many other kinases or other factors helping the stress granule behaving as a bistable system. So the CDK would not be the only one producing or giving stress granules this property. So this is just the result. So we think that for the stress granules and for many other systems, having bistable kinetics, well, could help them to become switches, and not just in this case for stress granule formation, but also for cell cycle arrest. Well, thanks for your attention, and please, if you have any question, I'll be happy to answer.
Thank you.